A very good morning, class. In today's session, I wish to give you an introduction to enzymes. We all are aware that enzymes are biomolecules which are very key players in the metabolism that is happening inside our body. So as biochemists, enzymes is a topic of utmost importance which requires understanding and also applies a lot to not only intermediary metabolism, it is also relevant to clinical biochemistry. Let's move on. So in this class, we'll try and understand the chemistry, the classification and the mechanism of enzyme action. Now going into its chemistry, we often refer to enzymes as biological catalysts that speed up the rate of biochemical reactions. Now, what catalysts can do to reactions happening in laboratories is what enzymes can do to the reactions that happen inside our body. Most of these enzymes are proteins and they are found to be globular proteins, obviously in their tertiary and quaternary structure. So we are all aware that there are 4 levels in the structural organization of proteins. The primary and the secondary that talk about the sequencing and the initial folding. But for a protein molecule to become functional, it should be present either in its tertiary or in its quaternary structure. Now in our days, we were often told that all enzymes are proteins, but all proteins are not enzymes. But now this statement doesn't hold good because they have discovered a special RNA species that is also having a capacity to act as an enzyme. These RNA species are referred to as enzymes. Now, if you look at the structure of the enzyme, there is a particular portion of the enzyme which is of interest. That part of the enzyme is referred to as the active site. Now, the active site is a region of the enzyme that can bind to the substrate. It can also bind to the other factors that are responsible for the conversion of the substrate to the product. So since this portion has to be accessible to the substrate, it is obvious that the active site is lying on the outer surface of this 3-dimensional protein. A protein is a very large molecule that has been folded back to back. So there are some parts of the protein chain that remain inside the folded structure, and there are certain parts that remain on the outer side. So this active site is on the outer surface of the protein so that the substrate can go and bind to it. Now if you're wondering how large this active site could be, then the active site is said to occupy only 5% of the total surface area of the enzyme. So it is very, very small compared to the large size of the enzyme. In addition, this active site is said to have a specific shape, and that shape is given to the active site by means of the tertiary structure of the protein. So in case there is a problem in the sequence of the protein, and in case there is a change in the structure of the protein, then the active site will not be functional. So the enzyme itself becomes a non-functional molecule. So much is the importance of an active site to the function of an enzyme. Now, what does this active site have to do so much with the function of the enzyme? Now, inside the active site, you can further divide them into 2. You have 2 binding sites which are helpful to hold the substrate in an orientation that is most suitable for the enzyme to perform the catalysis. So the active site has a binding site and it also has the catalytic site. So the binding sites are helpful in holding the substrate in a suitable conformation, while the catalytic site goes ahead with the catalytic function. Right. Now, there are so many different types of enzymes that perform so many different types of functions. But based on the structure of the enzyme, they have been referred to by different names. Now, you have a class of enzymes called the simple enzymes. Now, simple enzymes are those which are composed only of proteins. while you have certain conjugated enzymes. Now, conjugated enzymes are those which have a protein part and they also have a non-protein part. Now, the protein part of the enzyme alone is not enough for its function. The protein part, which is referred to as the apoprotein, has to combine with the non-protein part, which is called a coenzyme or a cofactor, in order for the enzyme to become functional. Now, if the non-protein part is removed from the enzyme, conjugated enzyme, then the enzyme is not functional. So, such a conjugate enzyme is also generally referred to as a holoenzyme. That is one which consists of a protein part and a non-protein part which is absolutely necessary for its enzymic functions. The non-protein part could be called a coenzyme or a cofactor. If it's an organic molecule, then the non-protein part is called a coenzyme. And if it is a inorganic molecule, then it is called a cofactor. Now, these cofactors and coenzymes, as you can see, they find a place to bind in the active site. So, they are not going to go and bind to some other place. They are going to be bound to the active site, the very area where the substrate comes and binds to the enzyme. So, without the cofactor, this proteinaceous molecule cannot behave like an enzyme. Now, these inorganic molecules that we often come across in intermediary metabolisms are magnesium ions, zinc ions. So, for example, magnesium ions are the cofactors that are required by the enzyme hexokinase, while zinc ions are the cofactors required by the enzyme carbonic anhydrase. Now, these organic molecules, generally when we say coenzymes, they are derivatives of the vitamin B complex protein. Vitamin B complex. Now, for example, glycogen phosphorylase, it requires a small organic molecule called pyridoxal phosphate, which is a derivative of pyridoxine, that is vitamin B6. Now, the coenzyme is rarely also referred to as a prosthetic group. Now, what is the difference between a prosthetic group and a coenzyme? Now, usually when a coenzyme or a cofactor is associated with enzyme action, their binding is reversible and they are loosely bound to the active site of the enzyme, and therefore they can be easily detached from the enzyme structure. But what differentiates a prosthetic group from a coenzyme is that a prosthetic group is again a non-protein part, which, like a coenzyme, is required for the action of the enzyme, but it cannot be detached easily from the enzyme structure. So when you try to remove it from the enzyme structure, you end up disturbing the enzyme structure. So that is what is the difference between the prosthetic group and the coenzyme. Now, next important part in an enzyme reaction is a substrate. Now, what we call as a reactant in a chemical reaction is what is known as a substrate in a biochemical reaction. So, for a substrate to get converted into a product, the most necessary step, the first step, is that the substrate has to come and bind with the enzyme and at the active site. So, the combination of an enzyme and the substrate is called as an enzyme substrate complex, which is the necessary first step for an enzyme catalyst reaction. Now, enzymes, like all other proteins, are synthesized by the ribosomes. And we know that ribosomes can either remain free in the cytoplasm or they can be attached to the endoplasmic reticulum. And the information regarding the sequence of amino acids in the enzyme is carried by the DNA. So, our cells can synthesize the required enzymes based on the genetic information that is coded in our DNA and use our ribosomes to synthesize these specific enzymes depending upon the requirement of the cell. Now, there are some enzymes which are referred to as intracellular enzymes. So these can be synthesized and they are retained inside the cell itself for their function. But there are certain other enzymes that are synthesized inside the cell and they are secreted from the cell outside to perform their function. The most common example that I could quote here is the digestive enzyme that is secreted from the pancreas. So these enzymes usually are transported to the duodenum where they perform their function.